So good morning, everyone. Sorry for taking time to set up this meeting. My laptop had some issues, and you can even see it. And I'm in Bomet. I'm attending a conference for the South Rift RCWG. Kelly, can you hear me? I hope you can hear me. Anyone?
Yeah, we can hear you.
Okay, thank you so much for being patient. Actually, I want to hand over this meeting to Ken, who will moderate on my behalf, because I need to go back to the conference. So we will I will listen to the recording, and of course, Ken is very able. He will be able to take us through the moderation, and Elfas will take us through the facilitation. So kindly allow me to hand over to Ken, who is here with me. Ken.
Good morning, everyone.
And you're going to move slides. You can't hear? For hiyo meeting, just toka tutumie tu hii moja. Juu itakuwa na echoes. Yeah, let's leave that. You can decide to. Okay. Now, so you can proceed. So you can proceed.
Presenter
Ni Elfas. So you can proceed.
Hello, good morning. Hello.
Good morning.
Welcome to today's presentation. This is Ken Omonya. I'm a clinical lead mentor supporting Kibra sub-county. So before we start, we can have a volunteer to give us a word of prayer, and then now we start our presentation. So anyone to volunteer kindly for prayer?
Let's pray.
Yes, let's proceed.
We are praying. Okay, let's pray. Our Father in heaven, we thank you this morning because of your mercies and grace that has enabled us to meet yet again. Father, in the name of Jesus, forgive us our sins. Cleanse us from all unrighteousness, Lord, so that we may be fit to serve you. This moment, Lord, we start this session. We ask you to be our guide in everything. And we ask you, Lord, that you take us through for the benefit of ourselves and our communities at large. We pray all this, trusting and believing in the mighty name of Jesus. Amen.
Thank you for the prayer. So now it is my time to welcome our presenter, Elfas Okapesi, to take us through the presentation of the day.
Thank you so much. For putting up this opportunity and this moment. Are you hearing me clearly? So that I just need to be sure I'm heard.
Yes, you are clear.
Yeah, thank you so much. As our colleague has mentioned, my name is Elfas Kapesi. I work with the University of Nairobi, and I support molecular testing, sequencing, and bioinformatics programs within the molecular medicine and infectious disease laboratory. I will be taking us through just an overview of what is this thing called genotyping and genotypic and phenotypic drug resistance testing, the yeah, you can move to the next. Your slides moving. Yeah, as we strive all of us to submit better healthcare previously. Yes, we all strive to really give better healthcare to fellow human beings. Mine is precision and evidence-based kind of medicine should be the way to go. The days of maybe struggling and scratching our heads and really trying to imagine and all that, I think those days are gone. We have a great opportunity with us. Yeah, next. Yeah, so you may be asking ourselves, what is this thing or monster called drug resistance testing? In itself, drug resistance is just a reduced effectiveness of a medication when we try to treat our patients. And then the vital process used to determine whether a microbe, for this case, you are dealing with a HIV, has developed resistance to a specific drug is what we call drug resistant testing. It's actually a vital process that helps us to determine whether this particular virus has developed resistance to our regimen that are at our disposal in our various facilities. Next. Yeah, so you may be able to ask ourselves, what causes this drug resistance? And one of the things that we shall be discussing is we are talking about the gene mutations. And then we talk about selective pressure of our regimens. And then talk about societal factors, which may include but not limited to misuse of drugs. And then we have what we call gene transfer. Yeah, next. Drug resistance may occur at least we have about two main with the virus, but that particular virus is actually resistant to certain kind of medications. So we call it a primary transmission. And then we have what called suppressive levels of antiviral drugs and possible natural selection of mutant strains. Yeah, that's the second one, which includes partial adherence to regimen. If somebody is on and off with the drugs, it's not very consistent. And then number two, we are talking about suboptimal dosing of drugs. If this person is supposed to be given a full dose, this person is given a dose that is not complete. So number three, you're talking about the drug interaction. The way the drugs interact within a host, within our patients, can also be able to really cause a drug resistance to occur. And then we have what we call incomplete absorption in the GIT. In a case whereby the patients have got some problems within the GIT, you realize that absorption, complete, adequate absorption is not taking place, then you could be sure that there will be a way drug resistance may develop. Next. Yeah, it's just the same thing as the ability of the virus to continue to multiply in the presence of a suppressive levels of ARV regimen. You know, you put our regiment there, and then you also put the virus there. But the measure is that the virus continues to do what? To multiply. So it means there is a problem. We expect our regimen to hinder or to block, you know, the progression and the increase of the virus, but it's not able to do that. And now we have what called drug resistant testing assays that are now part of the HIV clinical care. So that's why we are saying, we are talking about precision. We now have these assays that are able to tell us that this particular regimen maybe is not working, or maybe it can actually be able to continue supporting. and is widely used to guide therapy for better viral suppression and improved human survival. So this is the reason why we have now the lab at our disposal, and so that we do not really scratch so much our heads. Yeah, next. There are types of drug-resistant assays I may just want to run through. One of them is phenotypic. This is just a measure of the extent to which an antiviral drug inhibits virus replication in vitro. The result from this phenotypic assay is a value that defines the concentration of the drug required to reduce growth of the virus by 50%. So these are assays that are done in vitro, and you bring a virus, a sample with a virus, and the concentration of that particular virus, viremia, should be not less than 1,000 copies. And then you put also a regimen there in certain concentrations. And then the inhibitory concentration, what we call IC, of 50% of the patient's virus is compared with the IC of the drug sensitive. That is the wild type. The wild type is virus that is said to have not mutated from the original genome structure. And the fold change in terms of that comparison is now defined. So if the IC inhibitory concentration of 50 of an individual's virus is greater than that of the reference virus for a particular drug, then the individual's virus has decreased sensitivity to the drug. So we talk about the relative fold change helps determine whether the drug should still be included in the RT regimen or whether it should be removed entirely. So that is what we call phenotypic, assays. It's a bit laborious. Takes a long time, and it is a share of cones, and that is why it is not used widely. The utility of it is really very low. Next. Yeah, next. Yeah, so we are saying it is complex, quite labor intensive. It's also expensive, and it has got also a very long TAT. Yeah, on the other side, when you talk about genotypic, we are directly going to sequence the protein coding regions of the genes for viral reverse transcriptase. We have certain regions that are very key. We have the reverse transcriptase region, we have the protease, and then we have the integrase region, which produces these enzymes. So the mutations identified via genotyping are compared with a list of mutations that are known to confer resistance to current antiretroviral drugs. Yeah, so the moment we generate that kind of mutation in the in the pol region that we will be talking about, and then now we compare with already a list of mutations that are already known to confer certain kind of resistance to certain regimens. So we'll be going through the same, and this list is generated for RVs that are likely to remain active against virus versus those that to which resistance is likely. Yeah, so just a reminder to us, this is just an HIV structure and genome system. And our major focus right now, I know we have other regions of interest in which research is being done, but our major focus is coming down to the pol region. This is a region of our interest, and you can actually be able to see under the pol region, we have the protease region, we have the reverse transcriptase region, and then we have the integrase region. So those are the regions we are going to talk about even as we proceed. Yeah, next. Yeah, so the key enzymes of the pole region that drive the lifestyle or the life cycle of the HIV, and which are also very key in terms of drug resistance. Actually, three of them are reverse transcriptase, protease, and integrase. You will need to remember this even as we continue with the discussion. Yeah, so this is just a workflow. You can actually be able to see from a sample on your left as you move. We obtain that sample, and then we are able to do our RNA extraction, and then we move to conventional PCR, whereby we just amplify the RNA. And then after that, we want first to check whether there is whether there is a band. A band is an indication that there was amplification. Before now, we move to sequencing. And then thereafter, we generate a report containing the protease, the reverse transcriptase, and the integrase, even as we will be seeing shortly. Next. Yeah, so this assays the utilities purely just identification of the HIV-1 genotypic mutations associated with resistance to nucleotide and nucleoside reverse transcriptase inhibitors. And then we also have protease inhibitors and then integrase strand transfer inhibitors. And this particular assays can also guide in terms of initiation or change of combination therapy. So you can remember those two. So to us, when you begin the journey to manage this particular individual, we have a request form. And that request form has got just a client name and just basic biodata. And then there is a component, what we call clinical history. We expect you maybe to capture the regimen, the start date of that regimen, when was this switched, and then on the right side you capture also the viral loads in that order from the very first one, as you can actually be able to monitor the viremia versus the kind of regimen this particular client is in. So we expect you to fill this adequately so that it can actually be able to guide our investigation, even as we continue to give results. Next. Yeah, so in terms of the specimen involved in HIV drug-resistant testing, the primary sample is whole blood. So you just have to talk about whole blood. And then the container of choice we use now is the plasma preparation tube, PPT. This PPT has got a gel. So ensure that you have the PPT, which is not expired, with you. And the collection and separation, we advise within 2 hours after collection, you have to spin this sample. You separate the cells from our target, which is the plasma, because longer hours without separation can actually be able to lead to the virus exiting the plasma, the liquid phase of the blood, back to the cellular phase. So by the time you ship that sample here, we will realize that whatever viremia the patient has gone home with is different from the one you have submitted to us. And this assay depends purely on the presence, on the availability of the viremia in that sample. Centrifugation, 3,000 RPM for our lab colleagues who are here. 3,000 RPM for 5 to 10 minutes is sufficient to allow adequate separation distinct separation, whereby the cellular phase comes below the gel comes in between, and then the plasma, which is our target, now goes on top of the gel. And of course, after that, we will be able to get the plasma, EDTA, and then the volume that we estimate. If you use a PPT, that's about a 5 ml tube. That's a 5 ml tube. So when you collect so if you remove the cellular phase, so we can remain with about 1 to 2 ml, which is a very good volume. And then for the purpose of our clinicians, ensure that this particular individual you're investigating has not less than 1,000 copies per ml of the virus. And that 1,000 copies per ml has to have been done at least 30 days ago. I've seen requests for a client that was failing treatment, had about 5,000 copies 7 months ago. So if you bring just a sample for HIV-DR without really confirming a month ago, there is a possibility that the 5,000 may have become LDL. So anything LDL, anything below 1,000, it is really a very big challenge in terms of drug-resistant testing. We have also what we call specimen stability. It's important for us to note what kind of cold infrastructure do we have in our facilities. If you have -20, ensure the sample is in -20 for not more than 5 days. And if it is refrigerated, if you have only a fridge within your facility, ensure within 24 hours that sample has been shipped to the testing lab for the HIV-DR testing. And then, of course, as you're shipping, ensure triple packaging so that the sample and the cells do not mix, and also to avoid a lot of hemolysis. And then shipment, ensure you include the thermometer and then ship when it is processed. So that's just a basic information that can actually be able to go to our colleagues, those that are interacting with that sample from the word go, because the quality of that sample depends on the quality of the results we get. Yeah, next. Yeah, so these are just quality processes within geared towards supporting HIV drug resistance. And I'm sure our colleagues will be conversant with number one, just those are the supplies. And then remember, under supplies, you must have an HIV drug resistant request form, very key. And then number two, on this other side, you're talking about just a procedure for sample collection, properly label. It's important you label the sample with the CCC number, collection date, 5 ml, PPT, gently mix so that you don't bring a sample that has hemolyzed. The speed 3,000 within 2 hours for 5 to 10 minutes. And then down there, do not freeze that PPT before you centrifuge. Because anytime you, and I've seen this a lot within our lab, somebody was in a hurry, collected the sample, put into the freezer, hoping to come back in the morning and proceed with it. That's already a spoiled sample. You cannot centrifuge a sample because the pores in the gel shall have already compacted and they will not allow the cells to go through it. So as a result, because of the speed, you're going to have a lot of hemolysis now contaminating your plasma. Yeah, so ensure also ship the sample plus the request form. And if you're shipping the sample to us, kindly, that is the address. And then you are also encouraged, bullet number 4, you're also encouraged to make a call to the testing lab, because we all have a shared responsibility this particular client, from your side as the initiator and from my side as a provider. So make a call so that we can actually fast track, that sample. Take note of samples that fail to amplify. In many cases, when people send samples, they just, wait for results. And the result they expect is an HIV-DR report. But there is also another report that also comes when that sample fails. Yeah, next. So we now want to get into this process, just a process to determine arrangement and sequence of nucleotide bases. This is what we call genotyping. Talk about adenine, cytosine, guanine, and then thymine in a DNA molecule. Sequencing the whole genome of an organism is usually complex. That's why for this procedure, we have a Sanger sequencing that is going to do this, because it's going just to sequence a portion of the genome. And the target genes are broken down in the process. Sequence and small fragments are assembled into a single long, what we call consensus sequence. So it's a rigorous process that happens before we see the results. Yeah, so this is what happens. We have our client on the left. I collect the sample. This client is HIV positive and is confirmed as failed treatment. And then that sample, we extract it, and then we put them into the thermocycler. The extract plus the master mix, we amplify. And then after amplification, now we proceed to sequencing. And then, of course, within here, we have what we call the first PCR and the second PCR. And then we do the gel. The first PCR is what we call, for scientists who are here, reverse transcriptase, reverse transcription, because the starting material was RNA, so we have to reverse transcribe it into cDNA, and then that is now the cDNA we can proceed with. And then the second PCR basically is just nested PCR. The first PCR, we amplify the entire pol region, but in the second PCR, we narrow down either to protease and RT region only or to integrase region only. And then after that, we do purification, and then we can proceed and do sequencing, and then sequence reaction after purification. And then thereafter, we load to the sequencer where we have capillary electrophoresis, and then we obtain our sequence. Next. Back a bit. Okay. Yeah, next. Yeah, there was a slide that's supposed to come before this. So when we generate, when we get our sequences, yeah, this is what we get. Yeah, this is the output that we get from the equipment we are able to upload. There are two bioinformatics tools that we use. One of them is called Recall, previous slide. Yeah, so this is a Recall for the purpose of sequence analysis. We are able to get our results from the equipment, and then we are able to submit to this tool. And then after we have submitted to this tool, it generates to us the FASTA files. Next. Yeah, so it generates to us this file. So, and this, you're able now to check. Yeah, so after you have generated this one, now you can proceed to the next slide, which is now Stanford. This is Stanford. We paste it here. And then after this, we now generate our report. Next. Yeah, so this is just the process of pasting, and we are able to submit our FASTA files here, and then we are able to generate and analyze our report. Next. Yeah, this is now what we can come up with. Drug resistant interpretation protease, talk about the mutations, talk about the effect on the drugs, and you know, what happens now after we have done the sequencing. So this is the output, and I'm very sure most of us could be conversant with this. Next. Yeah, so this is just a sample report, still continuing. This is the RT region report, and with its mutations. Next. Yeah, so basically, when you talk about drug resistance, we cannot talk about drug resistance minus mutations. Mutations are basically mistakes that a virus makes, because a virus, HIV virus, does not have what we call proofreading mechanisms, whereby if it had made a mistake by inserting a particular nucleotide in a wrong position, unlike other host human, you know, organisms, you realize that the virus does not go back and check what mistake has it done. And some of those mistakes are lethal both to the virus and to the host. To the virus, those quasi-species being produced, some of them can be wiped away by the host immune mechanism, and some of them make the virus incompetent, meaning that it can no longer propagate itself. And some of those mistakes that generate such kind of mutations are also lethal to the host, the human host. For example, this M184V, this is a mutation on position 184 of the RT region. And there is a replacement of methionine, signified by M, by valine, signified by V. So in that position 184, in the wild type, there was M. But now, because of mutation, we have now V in that position. So that alone has got a great impact on the regimens that were targeting this particular region of the virus. Next. Yeah, so, and that brings us to the subject of transmitted versus acquired mutations. When an individual receives a virus strain that has got mutation, so that particular individual, when you look at the virus population, you realize that there are those that are wild type, maybe signified by the white ones. On the left, when you look at those diagrams on the left, and then we have the blue, we have the green, we have the orange. So those are what we call, we can talk about some mutant strains that are available. So when an individual is infected, you realize, based on the competency of those particular variants or strains, you realize that on this side, the blue one, the mutation of the blue one was offering it an advantage over the host. So you find the wild type, you know, getting wiped out, and then the blue one remains So when you talk about treatment, if you're going to treat this client based on the wild type alone, you realize that by the time this particular client continues to have high viremia, is because we have a mutant strain that is multiplying very fast within the system. And then, of course, acquired a situation whereby the virus within a host, due to interaction with the drugs and all that, can now be able to mutate into another strain. So someone has to ask yourself, what am I dealing with in this particular patient? Is it transmitted or acquired? And most advanced countries now, when somebody is to be initiated on drugs, they do a baseline HIV drug list just to know what kind of virus, what kind of mutations are available in this patient, so that they do not start from nowhere. They start from somewhere, from a known situation. Yeah. Next. Yeah, there is also, in terms of the inhibitors we have, which are regimens, there is also the concept of genetic barrier to resistance. There are those inhibitors with a very low genetic barrier. Simply a single mutation is sufficient to confer high-level resistance to this particular regimen. And an example is lamivudine. M184V, it is just sufficient to knock it out. You can no longer use it. And then we have K103N, conferring resistance to nevirapine and efavirenz, and also cross-class resistance. And it's also a very common drug-resistant mutation. So if somebody was having this kind of mutation, and this particular virus has passed to another person, I mean, the same kind of mutations will be transferred to the next person. And you may not really be able to use these drugs even in the next person. But there is also now what we call a high genetic barrier, whereby these inhibitors for this particular virus requires several mutations for resistance to be conferred on such inhibitors. These are very potent inhibitors. So the virus must have several kind of mutations for resistance to be conferred. And an example is lopinavir, ritonavir in a protease region, whereby you have L76V, it has to come in contact with Q58E. We have also L90M, and then we have I54V. So where all that can be able to occur, such kind of mutations, it can now be able to confer resistance to this particular regimen. So as a clinician, maybe you might need to understand which is that is high, which is that has got a low genetic barrier, which one has got a high, which one has got a low genetic barrier, which one now is available for you to use. Is it a combination or what? Yes. Next. This is just interpretation, which is available to us. When you get a susceptible, it means that there were some codon mutations present in a patient's HIV strain, but have not been associated with any resistance to the specific drug. So that's why you say you're comfortable there, you're okay, your drugs are okay, and maybe you just need to have your patient at the hearing. And then we have what we call potential low-level resistance. Codon mutations detected have been associated with a possible reduction in the susceptibility to the specific drug. So you are talking about now there is something reducing the susceptibility. Yeah. So you have now to put your eyes right there. And then there is low-level resistance. Codon mutations detected have been associated with a reduction in that susceptibility to the specific drug. So that's now where you need to begin thinking at that point. And then now we have intermediate resistance, codon mutation detected, also associated with that kind of susceptibility to the specific drug. And then now, before now you move to, it's actually just an alert, giving you an alert when you get a report of that, having those kind of words, potential, low level, intermediate, you have to gear yourself to something that could be coming ahead, maybe in a couple of months or something. Yeah, so codon mutations detected have been associated with a maximum reduction in susceptibility to the specific drug. So anytime you have a high level resistance, you'll be sure that that drug may not really work alone. There is really nothing much. Yeah. So sequence data obtained, okay, in terms of the report you get, you will also get what you call failed result. So the sequence data obtained are poor quality, and to determine the presence or absence of resistance associated codon mutations in the patient's HIV-1 strain. You may get such kind of reports we send to you, and this may be due to polymorphism in the region of the sequencing primers. If you're targeting the pol region, the RT, the PR, and the integrase, we have a lot of polymorphism, and this interfere with the primer binding and subsequent sequencing reaction. So this is the dynamics that do happen. You cannot see with your eyes. This is what really transpires within the equipment and the interaction between the sample and the parameters we have within the equipment. And then we have what we call low viral load, which is also a factor. If we cannot have sufficient viral copies for us to start our journey of amplification, sequencing from, it becomes a big challenge. Next. Yeah, so here with me, we just have some cases. We have some cases here. I've interacted with this patient. So this is case number one. Case number one, unfortunately, could not fit into one slide. So we have the RT mutations. We have D67G, S68G, K70R, M184V, T215V, K219Q. And you can actually be able to see that those kind of mutations within the nucleoside NRTIs, it has managed to submit how many high level? We have 1, 2, 3 high level, we have low level, we have intermediate resistance. And then of course, under non-nucleoside, we have high level, we have nevirapine and favirenz, just because of the K103N. And then of course, we have also intermediate, and we still have some, those that are still susceptible. Maybe they are not really touched. And then when you look at the protease, the next slide, I want you to look at the protease now. When you look at the protease region, there were no mutations detected, and that's why all the regimens are susceptible. And then of course, on the other side of integrase, we have mutations, and those mutations have conferred resistance to high-level resistance to cabotegravir and elvitegravir and reltegravir. So because of those mutations, you can actually be able to see the high-level resistance that has been conferred to them. And then for DTG, because I look at this patient, when I look at this patient was on TDF, 3TC, and DTG. TDF, 3TC, DTG. So possibly this, somebody could be noting down in case we could have some discussion on this. So this patient on TDF, 3TC, and DTG. And DTG here is has received intermediate resistance, maybe in a couple of months or time. You can actually be able to see that maybe it can also have high level resistance. That is case number one. Possibly you can note it. Let's go to case number two. Yeah, so this is case number two. There was no resistance. We were able to detect for reverse transcriptase region, and all the drugs are susceptible, you can see. And for those that are noting down, this patient number two, case number two, was on started on ABC, 3TC, DTG. That was on 27th of May, 2023. Last year, May, DTG, 3TC, ABC. And by last year, August, this patient had 116,873 copies. Yeah, so 116,873 copies, and it's on ABC, 3TC, DTG. Now let's check what the protease region and the integrase region have. The next slide. In the protease region, there was also no mutation. And DTG, high level resistance. This person was started last year. Already, the virus is resistant to DTG because of those mutations. So the questions we need to be asking ourselves, was this transmitted resistance, or it was acquired resistance? I think those are some of the things we need to ask ourselves. And this sample was collected 8th of November, 2023. The person was started on DTG, was moved to DTG in May 2023. So about 6 months, high level resistance to DTG. And of course, with the rest of the other integrase inhibitors. Next. Yeah, so as we continue discussing that, I just want to rush through the pros and the cons of genotyping assays for us just to be aware. The pros, we are talking about genotypic testing can be used to detect mutations that are causing resistance on a current regimen. So there was no any other tool we can use for us to detect these mutations which confer resistance. And then number two, it can also help to conserve treatment options by showing the ineffective drugs within a particular regimen. You can actually be able to see from the previous cases, there are those drugs now that, they are now ineffective. You can no longer use them. So it helps us to really point out which are those that are now ineffective, and continued uptake of those may just lead to toxicity and all those situations. So in terms of the cons, genotyping assays have got a minimum viral load of at least 1,000 copies per ml to ensure that we have adequate molecular amplification. And it's also not possible to detect low frequency mutations, what we call minority variants, especially if they are between 20% and 30%. Those are minority variants because this particular assay is population-based, especially for the one we are using for the Sanger is population-based. So the greater the percentage of the variants, those are the ones that will be detected. So it becomes very difficult for us to detect these minority variants. You may, in some situations, you may be able to have them having like mixtures in a certain aspect, but it becomes very difficult. And then capability of only detecting currently circulating strains is only able to detect what is circulating within the blood. We have no information of what is archived. So if whatever that is archived is a drug mutation strains, it becomes very difficult for us to really know what is inside there. And then it is better at determining which drugs won't work than which drugs will work. So it can only tell you that these drugs won't work, but may not really tell us that these drugs will work. So we need now to work using other patient information and all those other things for you to combine them so that you can use them to manage that patient. Yes, next. Patient's response to therapy, it's important for us to understand that the proportion of patients here that is drug resistance, that is a very key determinant. The proportion of the patients here that is drug resistant. Which kind of viruses are those that are drug resistant? They have which kind of mutations? And then number two, we are talking about patient compliance with the prescribed drug therapy. If the patient is not compliant, again, that poses another challenge. And then patient access to adequate care. What kind of care, what attention the quality of the attention we give to this patient, how is it? And then we also have drug pharmacokinetics. Basically, how is this drug metabolized, absorbed, and all those, factors that take place within the GIT of this individual. And then we also have drug interactions. A drug disinteracting with the other drug may lead that particular drug to become more potent or less potent. And then we are saying consider this when you're making therapeutic decisions. Consider all these factors and many, many more if they're there for you to make therapeutic informed decisions for effective management of our patients. Next. Yeah, so I finish by saying this road appears to have come to an end according to somebody else. But somebody else will say, no, let me get to that corner where it seems to be the road is coming to an end. And I see for myself what lies ahead. So this is the journey we have when you talk about HIV genotyping. Yeah, thank you. Thank you, everyone. Thank you. I appreciate it.
Thank you, Elfas. We really appreciate for the elaborate presentation. As the region, we'll still work with you for this wonderful presentation and ensuring we empower our healthcare providers. So I think now we can go to questions. And I think we can start with the charts. I see we have two questions, one for clarification and one question, and then we can now invite members to have their questions so that we can respond to them. So there was a need for clarification from Osea. He was saying, I missed something. Is the sample supposed to be transported with a thermometer inside? And what type of a thermometer? Kindly clarify. And then there's a question. They're saying based on the need for at least 1,000 copies of viral load for genotypic assay, does it mean we can't do DRT for persistent low-level viremia? That is 200 to 999 copies of viral load. Back to you, Elfas.
Yeah, thank you so much. we have, for our colleague who has asked about thermometers, we have some thermometers that, I mean, can just measure temperatures like maybe from -20, -30. I might need maybe to get the specifications and then maybe share. So those are like circular like thermometers. After you have packaged your sample, we have talked about triple packaging, and then you insert. And of course, first of all, it's important you just get these samples from -20. Yeah, you get these samples from -20 if you had collected a while ago, but not more than 5 days at -20. And then you transfer them to your packaging, and then you insert the thermometer there. So the thermometer will also be reading -20 by the time you complete packaging your sample plus the thermometer inside. And then you can also note down that temperature reading, if it's -20, as a starting point. And then you ship the sample through whichever means you use, whether riders. And then by the time the sample arrives, I will also note down the temperature at which it has arrived. Yeah, so that is why we take note of that integrity of that sample. Second question, low-level viremia. There is a concern at the moment, entities looking for HIV drug resistance for low-level viremia patients. But as I had said, the limitation of the genotyping assay is what is preventing us from giving out many reports about low-level viremia. Because to a very great extent, you find you do extraction, you proceed, but when you reach to the gel stage, you find there is no band. You cannot proceed. There is nothing to sequence. Yeah, so that is the biggest limitation we have. So that's why we say that we must be able to extract something for us to use in the downstream processes of the HIV drug-resistant testing. So you may get one report, but the frequency, the proportion of the reports you get from analyzing low level is really very low. That's why even WHO came in with at least 1,000 copies per ml. Yeah, thank you.
Yes, Elfas, thank you for for responding to those questions. So we also have a question or a concern. So on the issue of mutation becoming tremental, let me expand so that I can read for you. So on the issue of the mutation becoming tremental to the virus itself, does it mean one can get cured just as a result of this kind of mutation? A question from Prince. I think you can respond to that, Elfas.
Yeah, thank you, Prince. So as I had mentioned, there was a slide that had projected this particular virus, especially when we are at the dispensation of mutations. Mutations generate variants, very many different types of variants. So you find a mutation for a particular variant. So, but that mutation is little. I mean, it cannot allow that mutation to continue propagating itself. For example, if it made a mistake on the reverse transcriptase region of the same virus, so if it had made a mistake that will make its reverse transcriptase non-functional, that will be the end of that virus strain, because it will infect a cell, yes, because of the GP120 that is at its envelope, and then it gets inside, but inside it cannot do anything. So that actually becomes the end of that particular, virus or virion. And that's why we say that particular mutation is detrimental to the virus itself. But if it had the proofreading mechanism, it could have corrected it. And then so that by the time it infects a cell, then it can continue normally. Yeah. I guess you have gotten it, Prince.
Thank you, Elfas. So I see Christopher. So we can have two questions from Christopher and then Patricia, and then you respond to those questions. So we can start with Christopher. Christopher Mugo, kindly unmute.
Okay, thank you. My question is when you said the viral load must be within 30 days, taking into account that sometimes the TAT time is more than 30 days. How do you go about that? And also, Chris, guide us on those who are LLV.
Come up again in terms of the 30 days you're talking about?
You said for you to be able to do the drug resistant, the viral load must have been done within 30 days. Eh?
At least 30 days, yes.
Yes, and the TAT time sometimes is more than that.
Oh, for the viral load?
For the viral load, yeah.
Sorry. Yeah, so in such a kind of situations, you may be able to get with your team, and then that sample can actually be moved to a lab that has got a short TAT. So because we are all working for the good of that patient, that patient becomes like our priority for all us as an entire program. So you can liaise with your leaders and see where can this sample be tested within a very short time. I know we have labs in the country that give TAT for within 3 days. You already have your results. So if it's within 3 days, you have already checked the viremia is persistently high, and then from that point you make a decision and then you request for the HIV-DR. Yeah. Yeah, thank you.
Thank you. We can hear from Dr. Patricia. Yes, Doc.
Thanks, Ken. Thank you, Elfas, for your presentation. I think my question is somehow related to Christopher's. I'm looking at the DRT RRI that we did last year. And when we were doing the criteria for DRT approval, we said that the patient needed to have had a valid viral load within the past 6 months that's above 100,000 copies, and must have had at least 2 weeks of DWI, with the goal of us ensuring that we have sufficient drug pressure. So I'm looking at the 30-day gap, and I'm looking at the DWI. Does this mean then when it comes to us seeking to have DRTs, we need to change our criteria for us to have had a recent viral load within the past 1 month, and also to ensure that we have done the daily witnessing so that we have sufficient drug pressure so that this DRT counts? How best should we go about it moving forward? Yeah.
Thank you, Doc. Thank you. I do understand that before you select the client now for eligibility, this client is supposed to have failed several. If you look at maybe, I don't know whether your criteria is only the last 6 months or it can also go beyond. For example, I have a case here. If I can just run through. 2022, case number 2, it had 246,000 copies. That is 2022. 2023, February, it had 93,571. So I want you to see, for this particular case, in October 2022, 246,861. In February, that is like how many months? About 5 months. February 6th, 2023, it had got 93,571. 6 months later, that is August 2023, it has got 116,870. So when you look at all that, if you're talking about 6 months, there could be, for example, if you had somebody 6 months ago had 10,000, there is a possibility 6 months later, if the regimens and the inhibitors are potent, we may have an LDL. So that's why we came up and said, instead of that sample for 6 months coming in direct into the workflow of HIV drug resistance, we thought that we introduce at least 30 days. You can just, in the process of you requesting for HIV-DR, kindly also just request for VL also, maybe at the same time. And then if it is LDL, of course, we just come back to you and say, please, sorry, it is LDL, kindly go ahead and manage your patient as such. It's just for the purpose of the HIV drug resistant genotyping workflow that we have introduced this 1 month, because you are sure within that 1 month, even if maybe somebody was having very potent, the virus may not, will at least capture something before maybe it is wiped away. Yeah, so it's just for the purpose of the HIV drug resistant sequencing workflow. Yeah, thank you.
Yeah, I think that's in order. I think requesting a VL with the DRT would work so that we ensure that the patient, because a number of the high VLs we are getting are likely because of non-adherence. So that one would be a way to do DRT if we are not sure of the adherence. So I think it's good that we ensure that we have done a DWI for the 2 weeks and we request a VL so that if it is LDL, we don't proceed with we request the DL with the VL with the DRT. Yeah, thanks a lot.
Thank you, Daktari. Maybe the last question, Mr. Elfas, at times we receive samples and then there is we read somewhere whereby they say samples that has failed amplification. So maybe if you can elaborate on what do you mean by amplification of samples?
Yeah, thank you so much. There was a slide that tried to explain that as opposed to us getting a straightforward report, we also have cases whereby we run the first PCR, reverse transcriptase, and then we do the second PCR, and then we do gel. Unfortunately, we go again and do extraction again. We do again amplification, first and the second, and then we do gel. So in such kind of situations, we have encountered very difficult situations of that kind. The sample is not able to amplify. If we cannot see a gel band, there will be no report for HIV-DR for that particular patient. And that's why you see you do receive such kind of reports. So we do advise, because there are different number of factors that can lead to that. Number one is what we have just talked about, that maybe the sample was brought and we did not have a chance to check it is viremia at that point or within a month. And then number two, we also have limitations of the assays all the way from extraction. It is share of limitation. It may not be 100% in terms of extraction. Then you go to the next stage like that. It has got all those kind of limitations that do or comprise overall limitation of this assay. So, but that's why we are now coming down to really to start eliminating one by one. If it is the issue of 6 months, we want to eliminate that so that we are entering into HIV-DR workflow with a particular copy numbers. And then now from there, maybe if there are limitations based on the assays and the technologies, then now we can now be able to say, okay, this is based on this. Yeah.
Thank you, Elfas. We really appreciate for your time, for the good presentation. May I welcome Dr. Patricia, the chair of Nairobi Kajiado Technical Working Group, to give us a closing remarks. Thank you so much. Welcome, Dr. Patricia.
Thank you, Ken, and good morning, everyone. We are grateful, Elfas, for this enlightening session. I know there are some things that passed me by. I tried to flow. I think even the explanation behind amplification passed and failed. I'm yet to be very clear on whether I, as a healthcare worker, have contributed somehow to the failure or the passing of the amplification. But I want to believe that as we continue making these requests and going through our client DRT results, we'll be further enlightened and we'll understand better how to interpret our DRT results and how to use them for the benefit of our clients. What I can urge us to do as service providers is to ensure that right from the beginning, we insist on the importance of adherence to our clients and do the best to our ability to put them on the right combination of drugs and at the right dosage when it comes to matters to deal with children. The more our clients are developing resistance to medication, the fewer options remain available for their management, which will affect their health, their well-being, and their productivity. So let's just work together to ensure that we are doing our best with the resources we have. I think times have become much easier now. Elfas has given us a phone number. He has given us an email. So if he's not responding to the email, we are aware that we can reach out to him directly through a phone call. And we are grateful for that. We also need to work together with our respective mentors, county and sub-county level, and CASCOs and ESCASCOs, so that we receive the necessary support for the sake of our service delivery. I want to thank each and every one of you that has taken the time to be here. And I'm hoping that for the subsequent CMEs that we have planned as the region in relation to DRT, that we'll continue to attend, and that it will make it clearer how to interpret, how to appreciate the role of DRT in the management of our clients. Thank you very much. I wish us all a good day. Over to you, Ken.
We are free to leave. Goodbye. Kongoi Misin from Bomet.
Bye. Thank you. Yalama.
Bye-bye.
Bye.
That guy is a test, huh?
Miss,
Mtesa anasema the way I respond that is being unaweza mute.